Our next speaker is from Caltech, Dave Tirrell, and David will be speaking today on selective proteomic analysis. And unfortunately, he just wiped out my David in the middle of proteomic analysis of cellular subpopulations and complex biological systems. Please, David.
Glad I could help, Dan. Thanks for the introduction, and thanks to all our friends at the Dreyfus Foundation for setting up this symposium. I'd like to begin with congratulations to Bob Langer. Bob's influence on our field over a period now approaching 50 years has been extraordinary. Through his own research, through his technology development, through his vision for the field as a whole, his influence on young people, his ability to attract and direct investment to important biotechnological opportunities, and of course, most visibly in our current circumstances, his role in the creation of Moderna, which is not only essential to management of our current public health crisis, but has elevated all of science in the public view. And for all of those things, Bob, we're grateful to you. The work that I'd like to tell you about in the next few minutes has been going on in my laboratory in one form or another for something like 15 years now. It grew out of a conversation with two colleagues in neurobiology, Daniela Dietrich and Aaron Schumann. Aaron and Daniela are interested in the molecular mechanisms that underlie the formation of long-term memory, and in general, in the molecular mechanisms of synaptic plasticity in the brain. It's been known for many years that in order to form long-term memories, we have to make new proteins. And in part through Aaron's work, it's known that some of that essential protein synthesis occurs in synapses in response to signaling at the synapse. And Aaron and Daniela would like to know, and this is the problem they brought to us roughly 15 years ago, which proteins are made in the synapse in response to which kinds of signals. Now, this is a difficult problem because there are already thousands of proteins in the synapse before the new signal arrives and triggers the synthesis of new proteins. And so you have to find a small number and small quantities of new ones in the presence of much larger numbers of proteins that you don't care about quite so much in this context. And so the cartoon version of what's going on is shown over on the left-hand side of the slide. These dots are all intended to be proteins. This is the synapse before the signal arrives. This is the synapse after the signal arrives. There's a new dot on the right-hand side, but it's a little hard to find because your eye is drawn to all of the pre-existing signal. And so what you need is a way to make those pre-existing proteins invisible. And if you can do that, it becomes easy to find the new one, figure out what it is, and then maybe ultimately we'll understand why it's there. And so ordinarily, when my colleagues come to me with their problems, I explain to them that there's nothing I can do to help them. But in this case, I thought we might be able to help because some years earlier, in collaboration with Carolyn Bertozzi's laboratory, we had developed this molecule shown at the upper left, azidohomoalanine, or AHA. It's a methionine surrogate. It competes with methionine in protein translation. And so this offered the possibility of pulsing into some cellular protein pool before the signal arrives, a little bit of AHA. If some new proteins are made, they'll pick up AHA. They then become clickable. One can put on some sort of an affinity tag. I'm showing here at the bottom of the slide a biotin reagent, but we do this now in many different ways. But the key idea here is that now you can separate the newly synthesized proteins from the old ones, and you can identify them by mass spectrometry. A simple idea, and of course there are many variants of these kinds of ideas in chemical biology. It works quite well. An easy way to show this is to look at a Western blot. This is done in cell culture. We've treated cells in this second lane with AHA, and then we've clicked on a biotin reagent, and we look for those newly synthesized proteins with an antibiotic antibody, and you could see that there are lots of them there. When you do the negative controls, either you leave AHA out in the first lane or you put it in, but you inhibit new protein synthesis, you don't see very much background. So the selectivity of the method is pretty good. What we were worried about, and what many people would worry about, though, is whether we're not only probing protein synthesis, but perturbing it. And when we first started doing these kinds of experiments, it was very difficult to address that question because there was really no other way to look at the population of proteins made during a particular time interval. There was no other time-resolved proteomic method available. But some years later, this method of pulse SILAC, shown on the left-hand side of the slide, was introduced by the Selbach group. It's a beautiful method for doing time-resolved proteomics. Some of you may be familiar with how it works. You start with a sample that has no isotope labels. You then introduce two different ones, which we refer to as medium and heavy. You do whatever it is you want to do to compare the two different samples. You mix them, you digest them, and then again, you go to the mass spectrometer and see if there are differences between those two samples. And so what we decided to do was to develop control samples where we would always put in methionine, and that would be the medium sample. And then in what we refer to as the sample, we would either treat with AHA or with methionine. And so the question was, are there proteins that you find only in the sample that you've treated with AHA that shouldn't be there because they're showing evidence of perturbation of the sample by the addition of AHA, which is being incorporated throughout the entire proteome of the cell? And so J.D. Baggart, who did these experiments, did them in HeLa cells. He's done them in bacterial cells as well. The results are about the same. So the first question was, how many proteins do you find only in the sample culture? Well, when you just compare two nominally identical cultures, as we do on the right here, methionine versus methionine, you find about 3% of the proteins are only in one of the two samples. That's just the sampling variability that you get in an experiment of this kind. When we compared what we did in an AHA versus methionine experiment, the result was indistinguishable. And so we're not finding proteins that we shouldn't be finding when the experiment is done in this fashion. So that's protein identification, but you might be worried that we would find artifacts in protein quantitation. Does it perturb protein abundance? Not just which proteins are there, but even if the right ones are there abundances being shifted? And so here we're looking at a quantitative experiment where we're looking at the heavy to medium protein ratio along the x-axis, and then a measure of the confidence with which we can say that that ratio is different from 1. So if you compare methionine versus methionine, we're looking at about 1,000 proteins here. Everything is clustered down near the origin where it should be. Now we do the experiment that I described to you on the previous slide. We compare an AHA sample versus a methionine sample. It looks spectacularly different. If methionine is completely replaced by AHA, of the 1,000 proteins that we find in the sample, almost 400 are changed in abundance. But you would never do the experiment this way. We're completely depleting methionine here. All of the methionine is being replaced by AHA. There's no reason to do the experiment that way. And so what we did instead was to leave some methionine in there, put in some AHA that could compete with it, and then we look at the result when we have, say, a 30-fold excess of AHA versus methionine, and now we go right back down to the origin again. And so if the experiment is done in what I would regard as kind of a sensible way, you still find 1,300 proteins in this sample. We replace about 5% of the methionine, and now we don't get artifacts either in identity or in abundance. So the method can be made quantitatively reliable. But you might be wondering, if pulse SILAC is available to do time-resolved proteomics, why would anybody ever want to do a BONCAT experiment and mess around with the possibility of these kinds of abundance artifacts? So how much does the physical enrichment of the newly synthesized proteins really help? And the answer to the question is given at the top of the slide. It helps a lot if you're looking for a protein population that constitutes a small fraction of the proteome. And the nicest example of this argument that I've seen comes not from our laboratory, but from the Kreisfeld laboratory in Heidelberg, where they were looking at a comparison of secreted proteins. They wanted to compare the proteins that were secreted by two different cell lines in culture. And so over on the right-hand side of the slide is what you get from a conventional pulse Silac experiment. And what they're doing here is just comparing two replicates of the same experiment, replicate number 1 and replicate number 2, where they're looking at these protein ratios of the kind that I just mentioned on the previous slide. If you do a pulse Silac experiment, you find about 20 proteins, and there's no correlation between successive experiments. And the problem here is indicated at the bottom of the slide. The serum proteins are so abundant that it becomes very difficult to find the secreted proteins. On the left-hand side, we see what happens when they added a BONCAT enrichment step. Now they find almost 700 proteins with very good correlation between first and second replicates. And so, if you're looking for a protein population that represents 3/4 of what's in the cell, you don't need BONCAT. But if you're looking for a small fraction of the total protein population, the physical enrichment step makes the mass spectrometry much more effective. Then we started to look at more complex systems. We started with cell culture, but the next level of complexity for the neurobiologist is to look at tissue slices, and in particular, slices through the hippocampus. These are typically a few hundred microns thick, and they keep the tissue architecture intact. And so I'd just like you to look at the bottom of the slide and compare the lower left panel in each of those two parts of the figure. The one where I'm showing the pointer now on the left-hand side, we've treated that sample with AHA and a fluorescent alkyne dye. You can see there's lots of protein synthesis going on in that tissue slice. And then over on the right-hand side, looking at the comparable panel, we've added AHA and a fluorescent alkyne dye, but we've inhibited protein synthesis, and you don't see much background. So the method works in more complex systems too. But this raised an interesting question for us, and that is the question of whether we can do a cell selective analysis. You remember that we're interested in neuronal protein synthesis, and neurons are not the majority cell in the brain. They represent perhaps 10% of the cells in the brain. And so we would like to be able to look directly at what's going on in neurons without having to sort through the proteins that are made in all those other cells. So the question was, could we direct our analysis to neurons in particular? You can't do that with AHA because AHA is activated by the wild type methionine tRNA synthetase, and it's incorporated into proteins made in all cells. So what we needed was to identify an analog of methionine or another amino acid that is not activated by the wild type synthetase, but would be activated by a mutant synthetase that could be expressed only in the cells of interest. So we've done this in a couple of different variations now, but one that's been particularly useful for us is to use this kind of stretched analog of AHA, azidonorleucine or ANL. It's not activated by the wild type synthetase, but we were able to make a mutant synthetase that activates it quite well. Over here on the right-hand side of the slide shows a crystal structure of the wild type synthetase with methionine bound at the active site. The yellow ball there is a sulfur atom in the methionine side chain. And what we found from a library screen was that mutation of 3 residues around that active site would allow the synthetase to accommodate the azid side chain very well and provides good activation of ANL. The synthetase that we use in most of our experiments, we refer to as the NLL synthetase, because these 3 residues were mutated to an asparagine and 2 leucines. The idea then is that the NLL synthetase would be expressed in the cells of interest. We would be able to label proteins made in those cells and do a proteomic analysis without having to deal with all of the proteins that come out of the cells that don't carry the synthetase. So we've used this approach in many different circumstances now. An early illustration was a labeling experiment in which we were looking at the infection of cells by bacterial pathogens. And this shows an example of where we have incorporated the synthetase into the bacterial cells. You can see them highlighted in green here because we're clicking on a dye that shows up as green in this image. And we're looking at a mixed cell culture, bacterial cells showing in green and mammalian cells showing in red. We do the labeling in the mixed culture, and what you can see is that the labeling is in fact nicely restricted to the bacterial cells. So here you can differentiate one cell type, the microbial cell, from another cell type, the mammalian cell. But there are more challenging problems that one would like to be able to deal with, both in the microbial world and in the world of higher organisms. And one of those situations that we've been thinking about a lot over the years is the phenotypic variability that one encounters in biofilms. This shows an example of an interesting and important form of heterogeneity in Pseudomonas aeruginosa biofilms. This is not from our work. This is from the work of one of our collaborators, Tim Tolker Nielsen in Denmark. And what he's looking at here is the capacity of Pseudomonas cells to tolerate antibiotic treatment. And so I would direct you perhaps over to the right-hand side of the slide where we're looking from the side at the kind of tufted structures that form in Pseudomonas biofilms. And this is a live dead staining assay where cells have been treated with polymyxin. The dead cells are in the center, the live cells are on the outside. And it's often said that the reason biofilms are recalcitrant to antibiotics is that the antibiotic can't get in. Well, that's clearly not what's going on here because it's the most exposed cells that are surviving. And so that suggests that there are phenotypic differences in these films that are contributing to antibiotic tolerance. So we wanted to devise strategies, and we've now done this in a variety of different ways that would allow us to look in a spatially resolved way at biofilms, but also in a way that's dependent on the cell state. And so, for example, one of the phenomena that we've been interested in recently is the phenomenon of iron acquisition in biofilms. And so we decided we wanted to try to look at the iron depleted subpopulation in biofilms. And to do that, we needed a promoter that would turn on only under conditions of iron depletion and that would drive the synthetase expression in those cells that are suffering that phenotypic condition. So what we chose here, and this is the work of Xinran Liu, we decided to use the PVDF promoter. This is a pyoveridin synthetase F, which is responsible for the synthesis of this formylated hydroxyornithine unit that's used in pyoveridin synthesis. The pyoveridins are the high affinity iron acquisition system in Pseudomonas. So Xinran put the synthetase under control of that PVDF promoter. And I'd like you to look at the center of the slide where she's looking at the susceptibility of cells to labeling. And this is shown by clicking on an alkyne tamra dye shown in that upper panel of the gels that we're looking at. So across the lane at the top, the pluses and the minuses indicate whether or not the cells are starved for iron. Wild type cells don't label at all because they don't have the mutant synthetase. If you deplete iron, we get good levels of labeling. If you put the iron in, the labeling level goes down effectively to zero. It's not quite zero, but it's very low. And then if you look at the spatial nature of that labeling, this is where the proteins are clickable, as shown by the TAMRA signal. This is a standard cellular dye showing the tufted structure in its entirety. And what you can see from the merged image is that we can localize labeling to these deeper iron depleted regions. And we've now developed a whole set of promoters of this kind that allow us to look at different phenotypic subpopulations in bacterial biofilms. Sometimes a single promoter is not enough. It doesn't have the specificity that you want. And so the next level would be to require that 2 promoters be turned on. We did this by making a split form of the methionine tRNA synthetase, such that the N-terminal fragment would be driven by 1 promoter and the C-terminal fragment by another. We would get reassembly of the synthetase only in the cells where both promoters were on, and that would allow us to direct labeling to those cells. This worked quite nicely. We were confident that it would work because Paul Schimmel many years ago had shown that the methionine tRNA synthetase could be split and could be reassembled in functional form. We collaborated with Joff Silberg at Rice to find the best split site. And we found several good ones, but a particularly good one splits the synthetase after position 247. In order to show that this idea can be made to work, we decided to use a microfluidic system where we would get well-defined spatial turn-on of each of the promoters that we were interested in. We're doing this in a simple Y-shaped system where we have the 2 promoters as susceptible to induction by IPTG and by arabinose. We first look at the input signals. We bring IPTG in from the bottom here, and we're using it to drive GFP. You can see GFP turned on in what is presented here as the bottom part of the exit channel. When we bring arabinose in from the top, we can turn on GFP at the top. And now we're looking in the bottom panel, not at GFP expression, because those are our input signals. We're now looking at the output signal, which is the clickability of the cells in that output channel. And what you can see is that we get clickable cells right down the middle where we have overlapping opposed gradients of both inducers. And so this gives us a higher level of specificity in our proteomic labeling. Most recently, we've been applying these strategies to higher animals, and there are other laboratories doing this as well. We've worked primarily in worms because of the proximity of Paul Sternberg's laboratory. Daniela Dietrich has continued to work in flies, and Aaron Schumann's group has implemented these strategies in mice. The first thing we had to show was that you could label C. elegans with AHA, and that works very nicely. And I think I'm going to skip the information on the right-hand side because of the time, and I'm going to jump to the next slide, where we implemented a worm in which we expressed the synthetase under control of the RAB3 promoter. That is a pan-neuronal promoter, which turns on in all 302 neurons of the worm. And now, rather than seeing the entire worm light up when you click on the fluorescent dye, you see the nerve ring, the nerve cord, and the ganglia at the tail. Kai Yuet, who did most of these experiments, has now made half dozen or so of these kinds of constructs, which allow us to do selective analysis of each of the different tissues shown here. Again, because of the time, I'm not going to talk in any detail about what Kai was able to do, but in applying the method to the pharyngeal muscle cells, of which there are 20 in the worm, Kai was able to identify more than 2,400 proteins in those cells. The most ambitious variant of the experiment uses a split synthetase, and here Kai was trying to label just 2 cells in the worm. the ash neurons that are responsible for allowing the worm to learn how to avoid substances that it doesn't like. The problem here is that there is no single promoter that is turned on only in ash. At least we don't know of any promoter that's turned on only in ash. So what we did was use a split synthetase approach using GPA13, which is expressed in ash and a few other cells, and SRA6, which is expressed in ash and a few other cells. But the only one that they share is ash. And so we thought we could restrict labeling to the ash neurons. And I'd just like you to look at the lower right image here, where we're looking at BONCAT labeling. You see two cells lighting up in the worm. Those are, in fact, the ash neurons, indicating that there's at least preferential labeling in the two target cells in the worm. We've not yet been able to turn this into a good proteomic analysis because of the challenges associated with enriching proteins from just two cells out of the roughly 1,000 cells in the worm. So that's the story that I wanted to tell you about. And I wanted to finish with thanks to my colleagues and collaborators over the years. I've listed their names there and mentioned some of them as we've gone along. We've had good support from NIH and from the Army Research Office. And I suspect many of our speakers today will show us some of their favorite pictures of Bob. This is my favorite picture of Bob because he's being interviewed by my daughter, who is a health and science reporter for CNBC. This was a very exciting opportunity for Meg. And I have to admit, it gave me some credibility with my daughter. And any of you who have daughters would recognize that that's of great value. And so that's another thing for which I'm grateful to Bob. So with that, I'll close with congratulations to Bob, and I'd be glad to take questions if there are. Thank you.
Okay, thank you, David. Are there any questions? See, I'm looking in the chat. No questions yet. David, I was just wondering also with the biofilm formation where you're using sequestration for iron, they always also form at a triple interface, usually air, solid, liquid, because they need oxygen as well. Has anybody tried to regulate oxygen in biofilm formation versus nutrient regulation? Or is that too tough a problem, basically? It's easier to tackle the way you are.
You mean with respect to actually eradication of biofilm?
Yeah, for eradication of biofilm, since there's so many diseases.
Yeah, it certainly is critical in disease. I'm not aware that an oxygen depletion strategy has been used, although, in principle, one could think about ways to do that. I'm not aware of that strategy, but it seems like it's worth thinking about.
Okay. And then there's a question about whether you have problems getting the dye into the biofilm tissue.
Not as far as we can tell. That is something that we were worried about too, but it looks as though we get good penetration of the dyes. And in fact, many of the regions that we are interested in are regions that are well away from the interface with the medium. They're often sitting directly on the substrate. That's where a lot of stress cells are. So for example, when we use the RPOS stress promoter to drive the synthetase, we see a labeling pattern that looks a lot like the iron depletion pattern. So we don't seem to have difficulty in getting the dyes deep into the film or getting the ANL deep into the film. Okay.
And then just another quick sort of related, in the polymixin experiments, is it homogeneous, or I've seen polymixin sometimes just get stuck to the lipid membrane, and then it's hard to actually image through a whole cell, or have you not seen that problem with polymixin?
Those were Tim Tolker-Nielsen's experiments, so I don't know the details. the point that I was trying to make was that there was good penetration through the film of the
Right.
polymyxin because the cells that were being killed were the cells that were in the interior. And so the notion that, this is a transport phenomenon, I think it's a little harder. It's a little harder to make that case in that experiment.
Okay, thanks, David.